Hello students, my name is Ajay Kumar Shivastav and I am senior faculty in Botany at Sri Chaitanya, Gosala campus. Today we are going to discuss the topic molecular basis of inheritance. Molecular basis of inheritance. It is the second topic of the unit genetics. In the first topic, principles of inheritance and variation, we have studied two major things. First thing is characters are controlled by unit factors, which were later called as gene. And second thing, how transmission of characters takes place from one generation to the other generation. This type of study is called as classical genetics, or we also call it as transmission genetics. Okay, but two more questions need to be answered where we did not get the answer in this topic. First is, which biomolecule functions as gene? Functions as gene means what is the chemical nature of gene? Because Mendel has said that gene is a physical unit. If it is a physical unit, it means it is made up of some chemical. So which chemical functions as gene? And second thing is, what is the mode? What is the mode of action of gene? Mode of action of gene means how a gene phenotypically expresses itself. So these two answers we will be getting here in this topic. Okay. Now talking about the biochemical nature of gene, genes are made up of nucleic acids. This was proved by experiments of various scientists that we will be taking up further in this chapter. Okay? So genes are made up of nucleic acids, and nucleic acids, as you know, are of two types, DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. This DNA functions as gene. So chemical nature of gene is DNA, while RNA has some other functions like messenger, adapter, structural, and even in certain cases catalytic function. Okay, but biology is full of exceptions. So in certain viruses, like TMV, RNA functions as the genetic material, but in general the gene or genetic material is DNA and RNA has these functions. And these nucleic acids are made up of nucleotides. Nucleotides. So either RNA or DNA, these are polymers of nucleotides. So our study today will start with nucleotides. Okay. Now let us see what is a nucleotide. Nucleotide. It has two components, a phosphate group, and a nucleoside. Okay, this is H3PO4. Now nucleoside again has two constituents. Pentose sugar and nitrogenous base. Pentose sugar is of two types. Ribose sugar, this is found in RNA, and second, deoxyribose sugar, this is found in DNA. So the type of sugar is the main criteria for classification. RNA has ribose sugar while DNA has deoxyribose sugar. Okay. Now nitrogenous bases. Nitrogenous bases are also of two types. Purines and pyrimidines. Okay. Purines are of two types, adenine and guanine. Okay, and both found in both DNA and RNA. Pyrimidines are of three types: cytosine in both, thymine only in DNA, and uracil only in RNA. So these are the various constituents of nucleotides. Again, see, consist of two parts, phosphate group and nucleoside. Nucleoside consists of two parts, pentose sugar and nitrogenous base. Then pentose sugar are of two types, ribose sugar found in RNA and deoxyribose sugar found in DNA. Okay. Nitrogenous bases are of two types, purines and pyrimidines. Purines are of two types, adenine and guanine, and both are found in both DNA and RNA. Then pyrimidines are of three types. Cytosine is common to both DNA and RNA, but thymine is found only in DNA and uracil is found only in RNA. So this completes our nucleotides. Now we will see each constituent of the nucleotide one by one. First, phosphate group. Phosphate group. It is H3PO4. It has three free OH group. You can write it like this: P OH 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 and O. Then second, pentose sugar. Second is pentose sugar. As I said, pentose sugar is of two types. First, ribose sugar. Okay, ribose sugar has OH at carbon 2. So this is the main difference between ribose sugar and deoxyribose sugar, and as I said, it is found in RNA. So let us see. H OH H CH2 OH. Now see, this is carbon number 1, 2, 3, 4 and 5. So you can see OH is present at carbon second. This is ribose sugar. On the contrary, in deoxyribose sugar, H is present at carbon 2. Okay, and it is found in DNA. So this is the only difference, children. See, in place of OH here there is H. Okay, so here at carbon 2, one oxygen is absent, that is why the name deoxy. So this is deoxyribose sugar, it forms the DNA. Okay. Third constituent of nucleotide is nitrogenous bases. Nitrogenous bases. They have heterocyclic ring of carbon and nitrogen. Okay, these are of two types. First, purines. Purines are double ring structure, double ring. They have one pyrimidine ring and one imidazole ring, and nitrogen is present at position 1, 3, 7 and 9. So purine, double ring structure, nitrogen present at position 1, 3, 7, 9. And you know these are of two types. First is adenine. It has amino group at 6 positions, so it is also called as 6-amino purine. I will make the structure, but I am not going to balance the bonds. Okay, just to see the position of nitrogen. See. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. So you see nitrogen is present at position 1, 3, 7, 9 and at position 6 amino is present, that is why it is also called as 6-amino purine. Second one is guanine. Okay. Guanine has an oxo group at position 6 and amino is present at position 2. So you can say 6 oxo 2 amino purine. We can draw it something like this. So you can see at position 1, 3, 7 and 9 nitrogen, at 6 position oxo, and at position 2 there is amino. So these are the two types of purines. Next let us see pyrimidines. Pyrimidines. We are starting with pyrimidines. They are single ring structure. Single ring, and this is the pyrimidine ring, and nitrogen is present at position 1 and 3. Single ring structure, pyrimidine ring, and nitrogen present at position 1 and 3. These are of 3 types we have already seen. We are starting with cytosine. See, 1, 2, 3, 4, 5, 6, and here there is amino group. Okay, so this is cytosine. At position 4 an amino group is present. This is common to both DNA as well as RNA. Then uracil. The only difference is that in uracil amino is replaced by oxo, and you know uracil is found only in RNA. So see, so this is uracil. Okay, and third one is thymine. This is found only in DNA. The only difference between uracil and thymine is that in thymine at position 5 a methyl group is present. You see here 1, 2, 3, 4, 5, 6. Okay. Here at position 5 when a methyl group is present, then it is thymine. That is why thymine is also called as 5 methyl uracil. This is the reason why it is called also called as 5 methyl uracil. Let us see. So you see at position 5 a methyl is present. So this is thymine. So children, with this we have completed all the constituents of the nucleotide, that is phosphate group and sugar, pentose sugar and nitrogenous bases. Now we will see how a nucleoside is formed, and then we will see how a nucleotide is formed. So nucleoside is nitrogenous base plus pentose sugar. The C1 of pentose sugar forms a bond called as N-glycosidic bond with the nitrogenous base. Now if the nitrogenous base is purine, then the bond is formed between C1 of sugar and N9 of the nitrogenous base. And if the nitrogenous base is pyrimidine, then the N-glycosidic bond is formed between the C1 of sugar and N1 of the nitrogenous base. As you can see here, this is the sugar and this is, I have taken a pyrimidine. Okay, now let us see. this bond. This bond is the glycosidic bond. Okay, so glycosidic bond is a bond that is formed between or linkage between the nitrogenous base and the pentose sugar. This forms the nucleoside. Now children, see a nucleotide. So nucleotide is nucleoside plus phosphate group. Okay, and here the OH of the C5 of the pentose sugar and OH of the phosphate group, a bond is formed between these two, and this bond is called as phospho-ester bond. Let us see. So if you see this much, this is our pentose sugar, and if you see the upper part, this is the phosphate group. A water molecule is released from here. Okay, so a water molecule is released from here, and a bond is formed, and this bond is called as phospho-ester bond. Okay, so you see in a nucleotide there are two types of bond. The pentose sugar is attached to the phosphate group through a phospho-ester bond, and it is attached to the nitrogenous base through the N-glycosidic bond. So if you are asked that how many types of bonds are present in a nucleotide, there will be answer will be two. A phospho-ester bond between the phosphate group and the pentose sugar, and a glycosidic bond between the nitrogenous base and the pentose sugar. So this completes the structure of nucleotide. Now let us see how nucleotides are joined to each other to form a polynucleotide. For this I am showing an example of a dinucleotide. See, this one is a nucleotide. Okay, this OH is attached to the fifth carbon. Okay, this OH of phosphate C5. Okay, and this OHC, it is attached to C3. This OH to C3 and this OH to C5. Another nucleotide, same, see, another nucleotide. Okay, these two nucleotides come together. The OH of this one and the OH of the next one that is attached to phosphate, between these a water molecule is released. Okay, a water molecule is released, and this results in a bond. This bond is formed. So if you see, this is the bond between two nucleotides, and it is involving two ester bonds. That is why it is called as phosphodiester bond. It means in a polynucleotide, the adjacent nucleotides are attached to each other through a phosphodiester bond. Okay, phosphodiester bond. For this, the OH of the carbon 3 of the first one and OH of the carbon 5 of second one, between them a bond is formed. That is why we say that it is 3' end to 5' end phosphodiester bond. So phosphodiester bond is formed 3' end to 5' end. And from here children, 2-3 very important things you have to see. This gives polarity to a polynucleotide, because you see the free OH here is attached to 3 carbon. So this end is called as the 3' end, and this free OH is attached to 5th carbon. So this end is called as 5' end. So a polynucleotide has two poles, a 3' end where a free OH is attached to C3 and a 5' end where free OH is attached to C5. Now you know the name of the nitrogenous base, you know what is nucleoside, and you know what is nucleotide. So let us see this chart. For RNA, you see nitrogenous base adenine, so nucleoside will be called as adenosine and nucleotide will be called as adenylate. or you can say adenosine monophosphate. Similarly, guanine, guanosine, guanylate, cytosine, cytidine, cytidylate, or you can also say adenylic acid, guanylic acid. Okay. Similarly, cytidylic acid, and you know uracil is present only in RNA. So, uracil, uridine and uridylate or uridylic acid or uracil monophosphate. Okay. Similarly, for DNA, you know the sugar is deoxyribose. So, simply you have to add deoxy, nothing else. Here, if it is adenosine, it will become deoxy. Okay. So, you have to just add these things. Okay. So, you will get, and you know thymine, it is present only in DNA. So, this will be found only in DNA and this will be found uracil only in RNA. So, this chart you have to remember. Okay. So, students, with this we have completed the structure of nucleotides. We have seen the types of bonds found in a nucleotide. We have also seen when a polynucleotide is formed, then which bond is formed. Remember, in a linear single stranded DNA, the number of phosphodiester bonds will be n-1, where n stands for number of nucleotides. So, if there are 100 nucleotides in a single strand, there will be 99 phosphodiester bonds, and if it is 100 base pair long DNA, then 99 here and 99 there, then it will be 198. Okay. So, nucleotides, they polymerize to form nucleic acids. So, in our next chapter, we will be starting with the first nucleic acid, that is DNA, its structure, its discovery and its salient features. So, students, for the time being, that's all. In the next class, we will again start with DNA structure. Goodbye and please be very, very safe.